Our project is a comparative computational project on Rosmarinus officinales, Curcuma longa, Syzygium aromaticum, Zingiber officinale, and Withania somnifera, studying the effect of plant-based metabolites on the binding affinity between the ACE2 receptor and the Prefusion spike glycoprotein. Severe acute respiratory syndrome, or SARS-CoV-2, is a respiratory virus that has directly affected over 23 million people worldwide thus far, dating back to its emergence in December of 2019. There is research regarding traditional herbs being used to ease the symptoms of the virus, mostly targeting inflammation in the lungs, but not much research has been done in regard to how these herbs could potentially reduce one's risk of actually contracting the virus. This virus enters someone's body and causes them to contract it by attaching to the active site of the ACE2 receptor. ACE2 stands for angiotensin converting enzyme 2. The normal purpose of ACE2 is to convert an inflammation inducer, angiotensin 2, into an anti-inflammatory agent, angiotensin 1,7. Either by denaturing or blocking the active site of ACE2, certain substances could potentially be able to lessen the chances of someone catching COVID.
Our research question is, would metabolites from the previously listed herbs computationally bind the strongest with the SARS-CoV-2 perfusion spike glycoprotein or the mammalian ACE2 receptor in Schrödinger maestro in silico model? And the purpose of our project is to contribute to existing data within plant-based medicinal research towards controlling the spread of SARS-CoV-2 by finding an efficient, affordable, and safe way to reduce one's chances of contracting the virus. Our hypothesis is, if the binding affinity between the ACE2 receptor and the metabolites and the perfusion spike glycoprotein and the metabolites are calculated, then the docking value will be higher on average between the perfusion spike glycoprotein and the metabolites, because this protein is larger than the ACE2 receptor and therefore has a larger active site.
In the Maestro 12.5 suite environment set up by Schrödinger, a software used for pharmaceutical, biotechnology, and materials science research, we tested various proteins with metabolites. Each metabolite was chosen from the PubChem database, and each protein from the PDB, hence them being a controlled aspect. The proteins, prefusion spike glycoprotein and the ACE receptor, were tested with plant-based metabolites from turmeric, cloves, ginger, ashwagandha, and rosemary. The docking score was the output of these bindings, and these scores were compared against our controls, the positive being a docking score of -10, meaning that the metabolites were docking very well, while the negative control was a docking score of 10, meaning that they did not bind well and were actually repelling the receptor area. Almost.
Here are the images of each of our metabolites and proteins in Maestro.
First, we imported the structures of the metabolites and the proteins from the Protein Databank, or PDB website, into Schrödinger's Maestro suite. Throughout all the processes, all default settings were kept unless stated otherwise, and the pH was kept to 7.4 to mimic the human blood. First, we performed lig prep on all the ligands, which in this case are the metabolites. This turns the model from a 2D structure to a 3D structure and prepares it to bind with other molecules. Then we perform protein preparation on the proteins, which is the ACE2 receptor and the glycoprotein. Protein preparation automatically modifies the protein by filling in any missing bonds and ensuring structural correctness. This is done so each protein can bond properly to the metabolites. Next, a receptor grid was generated between each metabolite and each protein. This prepares the metabolite-protein pair to be docked, and this is shown on the top left corner of the screen. Finally, ligand docking is used to dock the metabolite-protein pair, and it produces a glide score, or docking score. This program rotates the pair into a number of different poses. We kept our poses at a constant of 10 for each docking job, and this is shown on the bottom left corner of the screen. We used a program in Maestro called XP Visualizer to view the docking scores each pair produced. Finally, t-tests were performed between each metabolite and ACE2 receptor and the metabolite and the glycoprotein. We used t-tests because our study had two main groups of data that were being compared to one another. The null hypothesis for our experiment is that there is no statistical difference between the docking scores of the metabolites, and our alternative hypothesis is that there is a significant statistical difference between the docking scores. We found that there is a significant difference between the population mean of 16 out of the 24 metabolites binding with the ACE2 receptor and the spike glycoprotein. P was less than 0.05. This is shown on the graph. Overall, most metabolites bound to the spike glycoprotein the strongest. The metabolites that have a caret symbol have p-values greater than 0.05, which is also shown on the graph. We would like to note that karyophylline was excluded from the data because there were no poses present, so no docking scores were produced in Maestro. It's important to remember that these docking scores are not strong enough to induce these metabolites to have direct effects on SARS-CoV-2, as a strong docking score is around -10. Therefore, more research is needed.
As for some limitations of our experiment, our project is solely computational, meaning that the project does not take into account bodily functions and processes outside of the proteins being used, as well as the state of health of the individual. Some examples of components that cannot be included are diseases, allergies, and symptoms that an individual may have. Our computational project also makes it so that the experiment does not consider environmental factors that can affect an individual's likeliness of catching the virus, such as the prominence and amount of COVID exposure around an individual.
We were also limited by the potential consequences of other molecules and proteins in the body that were not inputted into Schrödinger. The software is not designed to mimic the entire body, so putting all the other factors and proteins would have been too much for the system to handle. In the future, this team hopes to conduct a round of tests consisting of docking the metabolites with the spike glycoprotein and then with the ACE2 receptor. Additionally, we plan to test more metabolites from current herbs being used to determine if they neutralize or have a stronger inhibition. In order to continue our research for more effective and alternative options of neutralizing or inhibition, we will test metabolites of various other herbs. To account for other environments the metabolites could possibly have a strong binding with, we will test the metabolites with other receptors present in the immune and digestive system for more related results.
As for the implications of this project to society, using herbs as preventative treatments of COVID allows for a more accessible option as herbs are relatively easy to find. Additionally, the herbs do not require any prior preparation. Many third world countries may receive vaccines at a later time, but by using these herbs as prevention, the citizens of these countries are able to protect themselves and others with a treatment that is easy to find in many locations. Additionally, this treatment would provide a way to control the spread of the virus until the majority of the population is vaccinated. Thank you.